Okay, perfect. Takže už nás je slyšet. Jste si jistá, že nás slyšejí lidi?
No, I don't. What?
Hey, people. Okay, people online, can you hear us, please?
Yes, we can.
Okay, great.
Great. Good afternoon. I would like today introduce Michiel de Groot, who comes from University of Ghent, Belgium. First, I must say that I have a postdoc on my grant, Danny Hellewaters, who started in my laboratory to study ladybirds and their fungal parasites 3.5 years ago. Before that, he graduated in University of Harvard. Then he was in my laboratory, then in University Purdue, USA. Now he is again back in his home country, Belgium, in Ghent. And he has established there a group of entomologists and mycologists who study relationships of insects and their small, tiny parasitic fungi, which are very common but overlooked. And so we continue our collaboration. And this summer, during holidays, I will have his two students, PhD students, Michiel de Groot, who will now present, and another student, Brianna Sanmaria, from USA, Florida, master student, and they will study these tiny fungal parasites on ladybirds, which I am studying all the time, years and years. So today it will be introduction to this parasitic relationship of ladybirds and fungi by Michiel.
Thank you, Alda. Good afternoon, everyone. Everyone, mostly online, I think. I'm Michiel, PhD student from Ghent, as Alda explained. I just started my PhD and I'm working on a fungal parasite on an invasive ladybird. Fungal parasite is Hesperomyces harmoniae, which you'll see. And the invasive ladybird is Harmonia axoides, which you'll also see. But first I will have a short introduction on why to study parasites at all, which probably most of you are biologists, so you will know this, but parasites are particularly understudied. There's very little of their full diversity is actually known to us. And this is partially because they're hard to study, because some of them are endoparasites, which are inside other species. They're hard to grow in the lab. But they're also like unpopular. So even among biologists, the nicer the animal or the plant or something looks, it gets them studied more. So there's more people on, say, dolphins than there are on fungal parasites. So that's one reason. Another reason is that parasites are particularly interesting from an evolutionary standpoint. Parasites very often have a strict one-on-one relationship with their host, which means there's automatically an arms race going on between the parasite and the host. A famous example from history is, for example, on the famous dinosaur, the T-Rex. They had a parasite inside their bones called Trichomonas. And this might have been, maybe some people say that it's one of the reasons they might have gone extinct as well. And dinosaurs, of course, aren't around anymore, especially not Tyrannosaurus Rex. But Trichomonas is still here in modern day dinosaurs, which is the birds. There's also a reason to study parasites from an ecological perspective, because food webs all incorporate parasites. And if you compare a food web anywhere with just hosts, then it's way simpler than having parasites in there, which of course for analysis of ecosystems is way easier to just leave the parasites out, which happens quite a lot in ecology. But of course, to get the full breadth of biodiversity within an ecosystem, you need to include the parasites. And it's very informative because sometimes parasites have effects on various layers in ecosystems. Another reason is that parasites are specifically affected by the changing world, with climate change, with urbanization, any anthropomorphic change. Because parasites are, of course, affected by extinction in two ways. They can go extinct themselves, but also their host can go extinct. So if their hosts can't live anymore, then the parasite will also go extinct. So they're particularly threatened, and because they're understudied and their biodiversity isn't very known yet, we might see parasites go extinct all the time without even knowing. Yeah, there's another reason, but that will come up in the Harmonia bit. So here's our host. This is Harmonia axarides, the harlequin ladybird. It's an extremely polymorphic, very big ladybird. It gets confused with all kinds of species everywhere quite a bit because of they have a whole variety of colors. But you might also know it by another name, which is the Asian ladybird, because it's originally from East Asia. And it's a particularly hungry ladybird. It really likes eating aphids, which made it very useful for horticulture, which meant that horticulturalists and agriculturalists in North America imported it to the United States, where they fed on aphids and protected the plants. So they were used widely as a biocontrol, but from there they escaped in North America as well as and later they also spread to Europe, South America, Africa, and even to New Zealand. And this is a problem because they are very competitive. They outcompete other native species of ladybird, and they also are intraguild predators, which means that they actually eat other species of ladybird. So they threaten the native species wherever they are. So right now, one of the things we want is basically a biocontrol for this biocontrol. And here's our fungus. This is Hesperomyces harmoniae. This is a fungal parasite on Harmonia. Hesperomyces harmoniae is part of the Labubenialis group of fungi, which are, even for fungi, I don't know how much you know about fungi, but most of the time you'll think of fungi as a mushroom with a mycelium underneath it. And the mycelium is made up of hyphae, and hyphae are a whole bunch of threads. That's the typical fungus. But Labubenialis don't have any hyphae at all. And one of the stranger, which makes them one of the stranger fungi groups around. And they're ectoparasites on a whole variety of arthropods, so mostly on beetles like Harmonia, but also on harvestmen and on millipedes and a range of hosts we might have not even discovered yet. Labubenialis have been known since the 1800s, but only recently have there been more and more being described, including without sequence data, so just morphology, and with sequence data. And here you can see an example of the Labubenialis that's still in press, which looks particularly like one of those dinosaurs. Labubenialis so are made of a little cone-like structure called a thallus. And whenever the thallus is disturbed by, for example, mating, or also by social grooming or insects touching each other in whatever way, they release a little ascospore, which is a very sticky kind of spore, which then sticks to the infected host. And this is a very efficient way of distributing. So what this also means is that Labubuenalis, you can also very often find them on parts of the body which correspond to the way their hosts behave. For example, here on Harmonia, you can see on female ladybirds, they'll mostly be in the back. And on male ladybirds, they'll often be on the belly because, well, you can see that's the way they mate, and that's the way the Labubuenalis get transferred. It's not just mating in winter. When they overwinter, they cluster together in huge groups. And after winter, you can often find them completely covered in Labubuenalis. So this is clearly a completely covered Harmonia. And this is how you will often find them in the first months of the year. So there's something weird going on with Hesperomyces. So with the Harmonia coming from East Asia, the Hesperomyces detected on Harmonia have been first described from North America. So we don't exactly know how they have spread across the world, whether they followed Harmonia across the world or whether they had several points of infection. So that's one of the things I'm interested in my PhD, which is what we want to know. So which factors influence infection by Hesperomyces virulence? And we can determine this through lab work, which I'll talk about, and field work, which I'll also talk about. Then whether Hesperomyces virulence infection also increases Harmonia's mortality, maybe in combination with other entomopathogens. It might be interesting to look if we can actually use Hesperomyces as a biocontrol for Harmonia. And then how did it spread around the world? So there's phylogeography going on there. How did it go from Asia to North America to Europe and the rest of the world? So what causes infection? There's usually a whole range of factors involved in species being in a particular place. And so you can look at the ecology, which is open or closed, or even urban or agricultural or old forest or any of those things. There's abiotic factors like the location, which influences temperature and humidity. Temperature, humidity, and other factors that are specific to one spot. You could look at the individual hosts, like does it matter if they're male or female, or which color morph they are. And you can look at how the labules are spread on a particular host, so where they are on the host and what quantity they have. Does that affect the infection nearby? So you can do this in the lab, and this is actually one of the reasons I'm here, to learn about the lab work with Harmonia oxyritis. So you can put Harmonia into an incubator. You can change the dials on temperature and humidity, and you can try to see if Hesperomyces will grow more when it's more humid and the temperature is higher, or maybe the other way around. You can determine that through experiments in the lab. The other way you can try to figure out what factors cause more infection is by worldwide registration, because clearly, like, Harmonia is almost everywhere in the world. So what you need is a lot of ladybirds from all around the world. And for that, you need a lot of collaboration around the world. So these are all people I've been in email contact with, at least. And some of which have already sent some ladybirds, some of which are definitely going to send ladybirds out. One of them is Maria Christou, who is from Cyprus, actually an island that is completely devoid of any invasive species whatsoever because they kill them on site. And together with Maria Christou, I'm working on the Citizen Science project because Harmonia, as a ladybird, is a very popular citizen science project subject, because everyone instantly recognizes, oh, this is a ladybird, and will take photographs and put them online. And actually, a few of the cases of the first spreading of ladybirds, for example, there was the first ladybirds detected in several countries in Central America, which were detected using citizen science, including iNaturalist, but also looking at photos on Flickr. And there is some detection of some parasites of Harmonia done in Hungary. And there is, like last week, there was an article published by people in Greece of the spread of Harmonia throughout Greece. And actually, Maria Christou is I think she's the third author on that paper, and now I'm working with her. So she's already been really busy. She's been doing all kinds of ladybird seminars in Cyprus already, even though her short-term scientific mission that she's working with me in is only starting in May. So she's very enthusiastic, but she's also part of outreach group on Cyprus, working on telling kids and just the general populace about invasive species. And together we made a searching chart for people for how to, first of all, how to recognize Harmonia axyridis easily, and then to see whether they're infected by Hesperomyces. And then we asked them to record their sightings and either send them to me or use their local citizen science website to register them, or send them to iNaturalist, which is worldwide. And this seems to be an important part of this, because a lot of people will just go and look at the ladybird and say, oh, that's a very dirty ladybird. But actually it has Hesperomyces, and we need to tell people somehow to recognize it as such, rather than just being a dirty ladybird. So she translated this chart to Greek, and actually last week I got it back, this chart in German as well, as someone is translating it to Russian, so we're hoping to spread it as far and wide as possible. And then we're gathering all this information from various websites, including iNaturalist. iNaturalist is the really big registration site, but also Waarneming and LNB, which are the big websites of the Netherlands and Belgium. There's the European Ladybirds app, which registers ladybirds in general in all of Europe. It's specialized to ladybirds. There's Bug Guide, which is North American, and we're trying to include as many different websites from around the world as possible. And then we're collecting all this information on a single website called beetlehangers.org. And on Beetlehangers, we're making a giant map of wherever Hesperomyces has been detected, and with as much information as possible, as much as people give us. And for now, it's mostly North America, but we'll expand it to Europe and make it worldwide level 1 registration. So the other thing we need international collaboration for is the biogeography. So I will be doing DNA analysis on Hesperomyces from all over the world. This is a chart of how Harmonia spread around the world, and we're trying to figure out how Hesperomyces exactly followed it in its path. But this is in very early stages. This is still kind of far away. So, as a last thing, I'd like for anyone to keep this chart in mind. Take a screenshot now if you can, and send us a few observations of ladybirds, especially those with Hesperomyces on them. And we're also looking for collections of ladybirds with Hesperomyces on them. So if you ever have time, now's the time to send us some ladybirds. Right. That was it. There we go.
Well, here in Kokomo Room, we are mostly people that are familiar with this project, but we have two visitors who may have some questions. Katka?
Yes, I actually would have questions. I was wondering about the photo which was completely covered in the fungi. Was the ladybird already dead, or how much they can handle? Like, how fast is it killing them?
We don't really know. Like, it's actually quite hard to tell. But a ladybird like that will just happily walk around for a while.
So how does it go? Like, how effective they can be used as the pest control, or like?
Well, that's one of the questions we had. There have been some other research in which determined that there was like an effect on mortality. But in the wild, it's really hard to determine because the parasites might also infect the oldest and the weakest ones already. So it's hard to like distinguish between those two things.
Which one?
Yeah, it was.
It's just jumping randomly. Yeah, it was up.
There it is,
Okay.
yeah. This is the one you meant. Yeah,
Yeah,
there you go.
So this one is still happy animal.
Well, happy is probably a bit.
About it being happy.
Yeah, no, it's probably not particularly happy. And also, like the labos themselves, they actually like drill a little bit into the entrenchment of the outer layer of the beetle. So it is probably actually harmful to them. We're pretty sure, but how harmful? Yeah, that's.
It is still alive. It can live probably for a shorter time, and it will definitely have much less progeny, if any.
And I think there's also an indirect effect because predators will more easily be able to eat it because it's less, it can less move.
It's less mobile.
Because the thalli grow in between the joints and things like that.
Yeah, like that. Good.
I have a question. So the Harmonia was actively introduced in the United States, but then spread to other places. Is it possible this happened through agricultural exports or probably in other ways?
Yeah, so I think this is partially they were introduced as biocontrol, but I think the invasive groups of Harmonia actually have been introduced very recently. So they just, I don't know the exact way they spread, but they might have just come along on boats because they're so common and other ways.
They are rather synanthropic, so during autumn they enter human houses and other facilities to hide for winter in good mild climate, microclimate there. And during this they can hide in some packages of goods which will be shipped to another country. So there were also intentional releases, but last years mostly it comes with goods unintentional because they hide in something in the human constructions and so on.
And do we have some people with the questions online? No reaction, they're there.
Okay.
I have a question.
Yeah.
Do you have hypotheses about Hesperomyces spreading around the world?
I think it.
Where did it come from? Where did Harmonia pick it up? Like that kind of thing. Do you know, or do you? What's your?
I think the main speculation is that it jumped host somewhere in North America after exploding in population size there. But it's hard to tell because it's weird to think about a parasite that isn't there in their native range and then suddenly having this much success in other places in the world. But yeah, it's. Yes.
It looks like last chance for the people online.
Okay, well, thank you.
Thank you very much. Thank people who attend at least online, and probably tomorrow they will announce tomorrow seminar.
Yes, tomorrow. I should remember it.
I don't. Who is speaking?
Was Phil, right? Phil.
Yes, Philip, about the ants. About ants is Philip, ant community recovery in the Ecuadorian Choco. Thank you.
Moc děkuji, Katko.
Jo.
Brian tady pak bude delší dobu, ale